As for us, Atlantis Bioscience, we focus a lot on curating solutions that support your progress from bench to build. So today's webinar is actually a perfect example where Dr. Ong will be using some of the very tools that we provide to delve into the fascinating world of cancer and apoptosis. Let's give a warm welcome to Dr. Ong Ching Yi. The floor is yours.
Thank you, Cheryl, for your kind introduction. Welcome, everyone. My name is Ching Yi, and I'm from Atlantis Bioscience. So today, I'd like to share with you on the topic of tuning into cancer to explore more on the substrate stiffness and also apoptosis detection in cancer research. Before I start the presentation, please allow me to turn off my video to ensure the smooth transition of the slide presentation. Okay. I hope everyone can see my slide now. So without further delay, I'll move on to explain how the substrate stiffness matter in cancer research. First of all, let me introduce briefly on the tumor microenvironment. The tumor microenvironment consists not only of tumor cells, but also of vasculature, extracellular matrix, ECM, stroma, and also immune cells. It has been increasingly recognized as a key factor in multiple stages of disease progression, particularly impacting the local resistant, immune escaping, and also distant metastases. The bidirectional interaction between tumor cells and the microenvironment can promote tumor survival and aggressive cell behavior, and this will be an important factor that influence the development of the frontline interventions in clinical oncology. ECM is one of the major components in tumor microenvironment. The dysregulation of ECM is a remarkable feature of cancer. Sometimes we call it as ECM remodeling. Matrix stiffness, also known as ECM stress, is a critical physical factor of tumor microenvironment, and it remains alternating during the carcinogenesis. This is mainly due to the activation of cancer-associated fibroblasts, expression of remodeling enzymes such as the MMPs, extracellular collagen accumulation, cross-linking, and also fibrosis. Different content and density of ECM and those metrics with varying stiffness. During the cancer development, malignant cells contribute to the ECM stiffness, and in return, stiffened ECM alters the characteristic of the cancer cells. Tumor-associated ECM has been considered as a newly formed network that is essential for the protection of tumor cells and also the promotion of tumor progression. ECM stiffness provides adhesion and mechanical force for the tumor growth and also affect different perspectives of the cell function, as you can see here, evading the growth suppression, resisting the cell death, escaping immune response, promoting invasion and metastasis, inducing the angiogenesis, and also act as a drug diffusion barrier. Studies have shown that cells can sense their physical environment in a process called mechanosensing. And the physical signals induced by the matrix stiffness are transmitted to the cell primarily by the integrins receptor family and trigger a series of mechanotransduction that will eventually result in the changes of the tumor morphology, proliferative capacity, invasion ability, and also affecting the sensitivity of cancer cells to various therapies. There is another publication discussing on how matrix stiffness could induce the EMT, epithelial mesenchymal transition, and promote the chemoresistance in pancreatic cell cancer cells. The figure on the right shows the progression of EMT induced by matrix stiffness. The recapitulation of the fibrotic rigidities promote the elements of EMT and shift the cells towards the mesenchymal characteristic. And this also induced the cells to form chemoresistant to paclitaxel. The expression of PD-L1 in cancer cells play a vital role in the invading mechanism of the immune checkpoint system. In this study, PD-L1 was proven to be activated to be regulated by the substrate stiffness, where the substrate inhibits PD-L1 expression. This shows that the actin cytoskeleton is involved in the mechanism of the stiffness-induced PD-L1 expression. From these studies, we can see that stiffness actually matters in a remarkable number of ways. Especially in the space of in vitro culture, substrate stiffness has contributed to the mechanical matrix characteristic which eventually affect the biological behavior of the tumors. The closeness of substrate stiffness to the actual tumor microenvironment is particularly vital in the research such as the angiogenesis, tumor cell plasticity, tumor dynamics, and also the mechanobiology of the tumor growth. Every cell type adapts best to a certain stiffness of the surrounding environment. The conventional tissue culture plates that we usually use in the lab is nearly 1 billion times stiffer than the body's softest tissue. Scientists have long been finding ways to grow cells in either natural or synthetic matrix environment to elicit phenotypes that are not expressed on the conventional rigid substrate. Have you ever wondered how does the cell look like in our conventional 2D culture using the flowers and well plates? Our collective understanding of many cell-based processes is derived from the experiment performed on the flat, unphysiological, stiff material such as the polystyrene or the glass. While the simplicity of this platform is attractive, cells cultured in this environment tend to display aberrant behaviors, such as you can see here the flattened shape, abnormal polarization, and also altered response to the pharmaceutical reagents, as well as the loss of differentiated phenotype. The general stiffness-dependent cell behavior has been well demonstrated in the second figure. The cells form branch capillary light networks when cultured in soft matrix, but form larger tube-like structure with lumens in a more rigid matrix. Therefore, culture systems that better mimic the biological environment are needed to bridge the gap between the conventional 2D cultures as well as the complex native in vivo experiment. The 2D and 3D cultures are our common methodology for the cell culture. Although the 2D culture is convenient and easy to use, some limitations such as the high matrix stiffness, limited cell-cell interaction, non-physiological relevance are undeniable. The 3D culture, on the other hand, can offer us low matrix stiffness, enhanced cell-cell signaling, and also more physiological relevance. However, they come along with a complex culture system and also downstream process. Whereas the semi-3D culture that I'm going to propose today is a 2D culture with soft substrate. And most importantly, it combines the advantages of both 2D and 3D culture. To overcome these challenges, Matrigen has developed an innovative product, SoftWell, which enables the scientists to study cell behavior in a transparent hydrogels with varying elasticity using standard cell culture formats. Besides, it provides control over a range of matrix stiffness for the research of a variety of cell types. SoftWell hydrogels are made of polyacrylamide. They are so thin that common cell-based assays such as the wound healing assay or the invasion assay can be performed as usual. The transparency of SoftWell also allows most types of the microscopic imaging. Usual cell culture activities such as media aspiration, trypsinization, and even cell scraping can be also done as the hydrogel is chemically bound to the well. Lastly, the cells are also restricted to grow on the hydrogel surface, which makes them readily accessible for the isolation and downstream analysis. SoftWell is a transparent hydrogel bound to the surface of the culture vessel. It's less than a millimeter thick, so you might not notice it's there, but a cell can feel the difference. Elastic modulus ranging from 0.1 kilopascal to 100 kilopascal are available. SoftWell gels are also available as non-activated, easy coat, and also collagen. The non-activated hydrogels can be used as low protein and cell attachment surface, or alternatively as a substrate that can be chemically activated using your own methods. Easy-coat hydrogel are activated to bind to any extracellular matrix protein of your choice, while the collagen hydrogels are ready to culture option since the hydrogel are already bounded to the type 1 collagen. There are also HTS plates, which is the high throughput screen plates. It's an option designed to screen the cell behavior across standard stiffness values. It is available in 96 well plate format, and custom layouts are also available upon request. This slide shows the different formats of software that are available. We have the format of multi-well polystyrene plates, multi-well glass bottom plates, polystyrene dishes, glass bottom dishes, plus and also standard plastic plates with removable cover slips. That's the end of sharing on the first topic. Now I'm going to move on to the second topic of today, which will be focusing on the detection and tracking of apoptosis events using the fluorescent dyes. Apoptosis is a cell's natural mechanism for programmed cell death. As soon as the apoptosis is signaled, changes will start to occur within the cells. These changes include the activation of caspases, which cleave the cellular components required for the normal cellular function, such as the cytoskeletal and nuclear proteins. As a result of caspase activity, apoptotic cells will begin to shrink and undergo plasma membrane changes that signal the macrophage response, causing the eventual cell death. There are two different pathways that lead to apoptosis, which is the well-known intrinsic and extrinsic pathway that correlate with the signal type. The intracellular signal are DNA damages, growth factors, and also cytokine deprivation, whereas the most common extracellular signals would be the death-inducing signals produced by the cytotoxic T cells in response to the cells that are damaged or infected. Both pathways converge at the executional caspase, which is the caspase 3 and 7. Apoptosis play a critical role in the development as well as homeostasis. It serves to eliminate any unnecessary or unwanted cells and is a very highly regulated process. The prevention of cancer is one of the main functions of apoptosis. In certain circumstances, uncontrolled increased cell division rate can form a tumor, whereas when normal function of apoptosis is inhibited in cancer, the loss of apoptotic control allows the precancerous cells to survive longer and give more time for the accumulation of mutation, which contribute to the malignancy of the cancer. Therefore, apoptosis has become one of the main targets in cancer treatment. Common strategies for the therapeutic targeting include the stimulation of the pro-apoptotic molecules, inhibition of the anti-apoptotic molecules, and also upregulation of apoptotic signals. In order to detect apoptosis events at different stages, we need to understand what is happening at each stage. The translocation of the phosphatidylserine to the exposed membrane surface is an early event of apoptosis, where it serves as a signal for the attack of phagocytic cells. Admission 5 has a high affinity to the negatively charged phosphatidylserine that can be used as a marker of early apoptotic events. Other than that, the collapse of mitochondrial membrane potential is also one of the key events in the early stage of apoptosis. Collapse of the mitochondrial membrane potential coincides with the opening of the MPT pore, which is the mitochondrial membrane potential pore. And this collapse will can be eventually detected by the potential-dependent mitochondrial dye, which I'm going to introduce to you later. During the mid-stage, caspase activity is also involved. We can make use of the caspase activation assay to track the apoptotic cells. During the late stage of apoptosis, DNA fragmentation happens. It can be examined using the tunnel assay. The The in situ staining of the DNA strain breaks detected by the tunnel assay and subsequent visualization using the light microscopy can give biological significant data about the DNA damage and late-stage apoptosis. Anisin 5 CF conjugates can be used to stain the surface of apoptotic cells because of its high affinity for the negatively charged phosphatidylserine, which is this molecule that you can see here. So in normal viable cells, the phosphatidylserine will be located in the inner leaflets of the cytoplasmic membrane. During apoptosis, these little molecules, phosphatidylserine, will be translocated from the inner surface to the outer surface of the plasma membrane, where it can be detected by the fluorescently labeled anisin 5. This time-lapse video shows the apoptosis detection of the staurosporine-treated HeLa cells stained with the anisin 5 red color, and also oxazole yellow, which is the green color dead cell nuclear stain. And all of the nuclei are stained with blue host dyes. Mitofield dyes are fluorogenic mitochondrial stains for the live cells imaging. Loss of mitochondrial membrane potential is a hallmark of apoptosis. The dye is membrane permeable and becomes brightly fluorescent upon accumulation in the mitochondrial membrane. Mitochondrial localization is dependent on the mitochondrial membrane potential. When the potential is disrupted, the dye will relocalize to the cytoplasm. In addition to the Mitofield dyes, Biotium also offers several classical dyes to measure the mitochondrial membrane potential. These include the JC1, TMRM, and also TMRE, which allows to measure the mitochondrial membrane potential. Moving forward to the nucleophile caspase 3 substrate. It is made by attaching a nucleic acid binding dye to the caspase 3 or 7 substrate peptide sequence called DEVD. This uncleaved substrate is unable to bind to the DNA and remain non-fluorescent. Upon entering the cells, once the substrate is cleaved by the caspase 3 and 7 activity in the apoptotic cells, it will then release the high affinity fluorescent DNA dye, which will stain the cell nucleus with bright and stable fluorescent signal. The nucleophile caspase 3 substrate do not interfere with the caspase activity, allowing the monitoring of caspase activity in real time. In contrast to other fluorogenic caspase substrate or the fluorescent caspase inhibitor base assays, nucleophile substrate can be used to detect caspase 3 or 7 activity in the cells without inhibiting the apoptosis progression. If you are open to opt for a multicolor experiment, nucleophile combination staining kits are also available. You can couple it with the anisin 5, Mitofield, as well as the red dot tool for the concurrent measurement of the caspase cleavage and the loss of membrane integrity of the necrotic cells. Tunnel assay is a common assay for the detection of apoptosis. In this assay, the TDT enzyme will catalyze the addition of fluorescent labeled DUTP to the 3' ends of the cleaved DNA fragments caused by the caspase activity during the apoptosis. It can be used for the direct detection of the fragmented DNA by fluorescent microscopy or flow cytometry. Other than the apoptosis reagents and kits mentioned in the previous few slides, we also offer cell viability and proliferation assays, dead cell stains, and also the kits to detect microbial viability. I'd like to introduce you one of our star products, which is the VioFlow SE Cell Proliferation Kit. This is to use for the cell division monitoring and also co-culture study. The VioFlow SE dyes are membrane permeable compounds that are initially non-fluorescent, but it will be converted to stable fluorescent conjugates upon entering the viable cells and retained in the cells. It can be used to track cell division using flow cytometry approach. With each cell division, the daughter cell will inherit half of the fluorescently labeled, allowing the number of cell division that occur after the labeling to be detected by the appearance of the successively dimmer fluorescent peaks on the histogram of flow cytometry compared to the cells analyzed immediately after the staining. So ViaFlow SE dyes can also be used for long-term imaging morphology and also identifying the cells in co-culture by microscopy. This is particularly recommended for co-culture study because the stains is non-toxic and it does not readily transfer between the cells. Live or dye fixable viability dyes offer bright and stable labeling dead cells, label stabling of dead cells, and most widely used for to exclude the dead cells from the analysis in flow cytometry and through microscopy. The live or dye is a membrane impermeable dye that is able to enter the dead cells that have compromised membrane integrity. The dye labeling is extremely stable, which allows the cells to be fixed and permeabilized without the loss of fluorescent and also dye transfer between the cells. The staining protocol has already been optimized to maximize the live-dead discrimination with minimal live cell staining in order to prevent the interference with the immunostaining. CF dyes are actually a series of highly water-soluble fluorescent dyes developed and optimized for the labeling of biomolecules such as the proteins and also nucleic acids. These dyes offer superior brightness, photostability, and also solubility when compared to other commercially available fluorescent dye. They consist of over 40 CF dyes across the visible, red, far-red, and near-infrared spectra. The CF dyes are available as a wide selection of products, including the super-resolution microscopy, antibody labeling kits, primary or secondary antibodies, and also their conjugates, and also available as reactive dyes. Besides that, we also offer a variety of cellular stains that can be used to stain different subcellular components or structures for different application. You can approach us for the advice of the most suitable stain for your research. That's the end of my sharing today. Feel free to type in the Q&A chat box if you have any questions. And also, you could also contact me at biomedicine@atlantisbioscience.com after the webinar if you have further inquiries or questions. Thank you for listening.
All right, thank you so much, Dr. Ang. That was truly a fascinating exploration on how to control substrate stiffness for better cancer research, as well as how we can use apoptotic dyes to better study apoptosis and the detection of apoptosis. I believe your insights were definitely valuable, and I know that it will be a huge help to everyone attending here at this webinar today. So we have people that are even students and all the way up to seasoned researchers. And before we jump into addressing the questions from the Q&A, so there are some, if you are figuring out how to ask questions in the Q&A, so this is what you can do. So if you don't see the Q&A button appearing on your Zoom menu, you might need to click on the chat button to locate the Q&A section, or you might need to click on the More button to locate the Q&A section. So as we are waiting for more questions to come in under the Q&A session, you can take your time to type in. So for those of you that are typing in questions, or those of you that do not have any questions, while we await for that more questions to come in, I do encourage you guys to connect with us at Atlantis Bioscience and also Dr. Ong on LinkedIn through the QR code shown here. So following us on LinkedIn, you can get the latest update on the newest translational tools, and we will also often have learning snippets, like learning tools and snippets on our social media on LinkedIn. So do scan the QR code and give us a follow. We'll really appreciate that, and we can get connected there. So if, like Dr. Ong mentioned, if you do have any questions that maybe you are shy to answer here, the option is you can click on anonymous, but you can ask questions anonymously, or else you can email Dr. Ong personally through her email address here, biomedicine@atlantisbioscience.com. All right, I can see that there are some questions coming in. Let's see, what are the questions coming in here? Okay, so there's a question here, which is, what is the storage and shelf life of the soft well? Dr. Ong? I think your mic is muted, Dr. Ong.
The storage of the soft well is actually 4 to 25 degrees Celsius for the easy coat and also non-activated one. So for the collagen coated soft wells, it is recommended to store at 4 degrees Celsius. And for the shelf life of the soft wells, it's 6 months from the date of receipt.
6 months from the
Yes.
date of receipt. So that's like the storage time.
Yeah, but then before you use the software, you have to keep it properly and keep it hydrated in the sealed bag provided.
Okay, understand. All right. I hope that answers your question, Napat Soren. So then we have a few other questions. So one of the question is regarding the caspase staining. Is the staining if the staining is caspase, is the staining caspase 3 or cleave caspase 3?
The staining is actually for the cleave caspase 3. So after the caspase cleavage in the apoptotic cells, the high affinity DNA dye will then be released and fluoresce upon binding to the DNA.
Okay, understand. All right. Then we actually, thank you so much, Dr. Ong. We actually have another question here is there any collaboration for fellowship or research paper publications for that? Dr. Ong?
Collaboration for fellowship. I think Yvonne is typing the answer now.
Okay. So regarding collaboration, so this one is more for, I would say, Atlantis Bioscience side. So Yvonne will be representative to help us answer questions like that. So I think that's for the questions. Any more questions? We still have some time for more questions to come in. Okay. There's an anonymous participant. So their question is, can we scrape off cells from soft well for downstream application?
Actually, scraping off cells is not an issue for the soft well, but it is recommended to scrape off the cell using silicone cell scraper and also with gentle. Yeah, so you have to do a gentle scraping of the cells.
So gentle scrap. What if they accidentally scrape the hydrogel? Will that affect the results?
Usually when we use a gentle scraping, the cells will just come off. So it's the hydrogel would be chemically bound to the well, so theoretically it wouldn't be scraped off.
Okay, understand. All right. Then I would say the answer is done. Okay, I think we have time to accept one or two more questions. Anyone have any more questions before we jump to our next section, which is our giveaway? Let's see. Any more questions? All right, okay, there's one more question that came in. How many cell division does the via floor dye dilution last?
The via floor dye dilution actually it depends on cell types. Usually would be 4 days. Yeah, within a week, I would say. But then the days would depends on the cell types.
Depending on the cell type.
Yes.
Okay.
So we also offer trial size kits. for optimization purpose. So if you wish to try out this dye, you can get the trial size kit from us first before you commit to a larger size for experiment.
Okay, so we do provide a trial kit for this biofloor dye lah. Okay, I hope that answers your question. So we have two more questions coming in. Which assay do you think is the best for the whole procedure of apoptosis? Only tunnel or other?
Actually, it would highly dependent on the stage of apoptosis you are looking for. So for certain drugs, if they are targeting on early apoptosis, then of course you will need to opt for the early apoptosis detection assays. Yeah. So there is no best one for study. So it will depend on the type of drugs or the end goal of your experiment. Yeah.
So Sumaya, if you want to follow up, you can maybe share with us more, like what you are trying to, what is the end goal of your studies? Then maybe Dr. Ong can help you more on that question. So then we have another questions from Nicha Khan. So this question is asking, do you have data applying Softwell in other disease studies, such as tissue regeneration?
Yes, Softwell is also recommended for a differentiation study and also tissue regeneration. I can share with you the publication later. Can you leave me your email?
So Chakan, you can put in your email. Maybe you can, after we click on answer it, then you can put a comment of your email so Dr. Ong can reach out further to you with the other applications and other studies. All right, Sumaya said, yes, Sumaya said he's thankful and he got it. So I think that concludes our Q&A session for today. So if you do have any more questions, again, feel free to go and text Dr. Ong here, or if you happen to miss out this email, you can also actually message us on LinkedIn or through our website. So there are many channels out there. You just need to find Atlantis Bioscience, then you can get to us.
Fluorescent probes and their ability to label biomolecules have served as the backbone of life science research for many years. Yet commonly used fluorophores such as PHYTC, SiDyes, and Alexa fluor dyes often suffer from significant performance issues, including high background, low signal, poor photostability, and a limited color selection. Introducing CF dyes for clearer fluorescent results. Biodium's novel fluorescent probes that are chemically engineered to offer the best possible performance for your research. CF dye labels are available in over 40 colors across the visible and near-infrared spectrum, and boast unrivaled performance in brightness and photostability. This is achieved through Biodium's patented polyethylene glycol, or PEG modifications. Fluorescent dyes such as Alexa fluor and SiDyes often contain negatively charged functional groups that increase non-specific binding and background fluorescence. The unique PEG modifications on CF dye labels shield these negatively charged functional groups, reducing background fluorescence as well as significantly improving brightness and overall performance of the labeled conjugate. As a result, CF dye-labeled antibodies and other biomolecules offer much brighter signal and less background in immunofluorescence microscopy and Western blotting, allowing for more critical analysis. In flow cytometry, antibodies labeled with CF dyes offer exceptional brightness and signal to noise. In addition, The CF dye series offers the widest selection of spectrally unique dyes to accommodate highly complex panels for use in both conventional or spectral flow cytometry. Biodium scientists also developed CF dye probes specialized for super-resolution microscopy. The photostability, brightness, and photochemical switching properties of certain CF dyes make them ideal for storm, STED, SIM, and other super-resolution techniques. CF dyes also lead the industry in near-IR fluorescence. Using advanced molecular engineering, Biodium scientists have produced novel fluorescent probes with wavelength emission well over 800 nanometers, extending beyond all other commercially available fluorescent probes. These near-IR CF dyes offer superior brightness for the labeled conjugate. And due to the strong tissue penetration, low background, and linearity of near-infrared wavelengths, near-IR CF dyes offer significant advantages for in vivo imaging and other applications. CF dyes have been licensed by several leading biotechnology companies and are available in rapid and efficient mix and stain labeling kits, primary and secondary antibodies, widely used bioconjugates, and reactive formats. Let CF dyes for clearer fluorescent results enable your next breakthrough with a wide selection of ready-to-use probes for applications including microscopy, flow cytometry, and Western blotting. Visit biodium.com to learn more about CF dyes, as well as Biodium's full breadth of fluorescent technologies aimed at accelerating your research.